The principles of classical genetics, and in particular the discoveries of the Czechoslovakian monk Gregor Mendel, set the stage for discoveries at the cellular level. In Lecture 49, we're going to explore the seminal discovery in modern genetics that DNA, the double helix, carries the genetic code. In this lecture, I have two principal objectives. First, we're going to consider cellular genetics and the microscopic chromosomes. These are structures that display fascinating and suggestive behavior during the two different kinds of cell division called mitosis and meiosis. Then we're going to see how scientists discovered the chemical identity of the genetic molecule DNA. Recall Mendel's 4 laws of classical genetics. These provide a framework for all subsequent discoveries in cellular and in molecular genetics. First of all, there exist genes, and these genes carry traits. Secondly, each individual carries 2 genes associated with each trait, one from the mother and one from the father. Third, genes come in different forms, or alleles. They're dominant alleles, and they are expressed in favor of the recessive allele. And finally, Mendel said that different alleles are sorted and distributed randomly so that all combination of alleles are equally likely. Mendel's work, like many important scientific research, raised new questions in genetics. What is the physical and chemical nature of Mendel's atoms of inheritance, for example? Every biological system must carry information in these atoms of inheritance. They have to be physical structures. What are they? Another question, how is the biological information stored? How is it interpreted? How is it passed from one generation to the next? Fundamental questions in genetics. Also, how do alleles differ? How come they give different traits? How come some are dominant, some are recessive? New insights were provided by examining cells and the cellular structures called chromosomes. Cellular genetics is the study of the transfer of biological information at the level of cells. This research requires microscopes, of course, because cells are so small. They are microscopic structures, and that's why the discovery and the invention of the microscope and then subsequent improvements in the microscope was a critical part in this history of cellular genetics. This, of course, is contrasting with the work of Gregor Mendel, who worked in classical genetics, the study of organisms, which you, of course, can deal with in a garden. You can just look and see whether a plant is tall or short, whether it has yellow or green peas, and so forth. There were 2 key microscopic discoveries that set the stage for this work. First, of course, is that every living thing is made of cells. The cell is the fundamental building block of life. And the second is that all cells arise from previous cells. So if you're going to look for the biological information, you have to look for something inside the cell that is passed from one generation to the next. That's where the information has to come. While observing cell reproduction, scientists noticed striking behavior of elongated structures in every cell. These are called chromosomes. In cells with nucleus, these chromosomes occur in the nucleus, and you can stain them by various chemical techniques so that they occur in bright colors in the cell, and therefore they're more easy to observe when you look through your microscope. Certain characteristics of chromosomes were very suggestive. For example, all chromosomes occur in pairs. Another thing, different species have different numbers of chromosomes. For example, humans have 23 pairs of chromosomes, but some organisms only have 1 pair of chromosomes, some have more than 100 pairs. The chromosomes show very striking behavior during cell division, and this is one of the things that led geneticists to focus in on chromosomes. Indeed, as we'll see, there's some parallels between the behavior of chromosomes and those laws that Mendel proposed for classical genetics. We see that most cells divide by a process called mitosis, in which one parent cell becomes 2 identical daughter cells. And here's what happens to the chromosomes. Just before cell division, the chromosomes thicken, and they arrange themselves into pairs. And each of these pairs looks like a little X, sort of 2 crossed chromosomes, if you will. These X-shaped pairs of chromosomes then line up as if they're along the equator of a nucleus. So you think of the nucleus as a globe, and the pairs of chromosomes, those X's, line up along the equator. Meanwhile, a bundle of fibers appears, and these fibers stretch from one pole to the other pole of the nucleus, sort of like longitude lines on a globe. What happens then is those X-shaped pairs of chromosomes are pulled apart. They split along the longitude lines, and half the chromosomes go to one pole, the other half of the chromosomes go to the other pole. Then, two new nuclei form where before there was only one. A nucleus forms at one pole, another nucleus forms at the second pole, a cell wall forms along the equator, and eventually you have two identical daughter cells where before there was only a single parent cell. During this process, the chromosomes have doubled, and each daughter cell has a full complement of 23 pairs of chromosomes in the human, for example, or the appropriate number in other organisms. Now, there's a very different process of cell division that's observed during sexual reproduction. And these differences provide a key to understanding the genetic mechanisms at a cellular level. This process, by the way, now is called meiosis. I always remember it this way. Mitosis is when you produce two cells from one. That's the simple process of cell division. But meiosis is the sexual reproduction side. Now, each species has a characteristic even number of chromosomes in every cell. Humans have 46, dogs have 78, grasshoppers have 22. These chromosomes then occur in matched pairs. They're called homologous pairs. So, for example, a grasshopper with 22 chromosomes has 11 homologous pairs of chromosomes. That is every cell except for the sex cells, and this is where we get an interesting difference. Egg and sperm have only half the usual number of chromosomes. In humans, there are only 23 single chromosomes. So when the egg meets the sperm, the 23 single from the male, 23 single from the female, give you 23 pairs of chromosomes. These sex cells, the egg and the sperm, are called gametes. And two gametes combine then in the fertilized egg. Many discoveries about this process were made in 1902 by the American biologist Walter Sutton. He was born in 1877. He died prematurely in 1916. He is the one that discovered that the 11 chromosomes from the egg of a female grasshopper have a matching 11 chromosomes in the sperm of the male. So he made this fundamental discovery. This is why ordinary cells really do have the pairs of chromosome, half from the father, half from the mother. It's really sad about Sutton. He made this important discovery while he was still a graduate student at Columbia University. He then decided that medicine was going to be his career. So he published one important paper. It was called The Chromosomes in Heredity, published in 1903. It stands as a landmark in genetics. But then he went on to become a surgeon, and a very gifted surgeon apparently. But he died tragically at the age of 39 from a ruptured appendix. If he'd had access to a surgeon as gifted as himself, he would not probably have died from this ruptured appendix. Okay, let's talk about this process of sexual reproduction. To obtain a set of chromosomes, sex cells have to undergo a special process called meiosis. The first step is just like mitosis. That is, all the chromosomes divide, so if you start with, for example, 23 pairs, you then end up with 46 pairs, which arrange themselves in these thick X-like arrangements. And just as in mitosis, those X-like arrangements of chromosomes line up around the equator of the nucleus. But meiosis deviates from mitosis at this point. Spindles form just as in mitosis, but the X-shaped structures are not pulled apart to give new cells, each with 46 chromosomes. Instead, what you find is that 23 of those X-shaped pairs are pulled to one pole, and 23 of the pairs are pulled to the other pole. So there's quite a subtle distinction here between meiosis and mitosis. And you might want to look in the booklet to see the illustration of how these two differ. Then another set of spindles forms. And the two cells, each that have the 23 pairs, now split into 4 cells, the 4 gametes, the 4 sex cells, each of which now has 23 single chromosomes. In the female, they're the eggs. In the male, they're the sperm. And then again, at the moment of fertilization, 23 chromosomes from the male are combined with 23 chromosomes from the female to give a new, unique combination of genes, half from the mother and half from the father. Let's think about gender in this process. A close examination of chromosomes shows that human males carry one pair of chromosomes that's not exactly matched. That 23 pair has a longer chromosome called X and a shorter one called Y. All males have this XY combination, whereas all females have the XX combination of the two longer chromosomes. Now, this seems to be the only genetic difference between males and females in the human species. So all of the male sex traits have to lie on that Y chromosome, which is unique to males. So do other sex-linked traits, things like baldness, color blindness, which is much more common in men. These are traits that must also lie somehow along that short Y chromosome. So think about what happens during the production of gametes. In females, every egg has to carry only X chromosomes because that's what a female has, is two X's. But in males, half the sperm carry X, half the sperm carry Y. So when fertilization occurs, the decision of whether or not you have a male or a female is really up to which sperm happens to fertilize the egg, whether it's an X or a Y carrying sperm, and that's why there's a 50-50 chance on average of having a boy or a girl. I was amazed to learn that other species have a different strategy, that this isn't universal among sexual reproduction. Female birds, for example, are XY, and the males are XX. So in birds, it's the female, the egg, that determines whether or not you have a male or a female offspring. And then in some insect species, there's a completely different pattern. In some insect species, all males develop from unfertilized eggs and therefore have only a single sex chromosome. It's only the females that have two sex chromosomes. Different strategy altogether. And then there are quite a number of species which can switch back and forth from male to female depending on demand, depending on environmental factors, for example. There are a number of fish and a number of amphibians, for example, which in a population, if too many males are around, some of the males will spontaneously switch to becoming females. And they can switch back again later in their life. So they're amazing strategies, and the human is only one representative of the different kinds of ways that genetic information is passed on and that gender is determined. A major challenge facing biochemists of the early 20th century is to discover the relationship between genes and the biomolecules that make up an organism. After all, genes somehow have to provide information on how to build chemicals. They provide coded instructions, information, if you will, about how the chemical laboratory of each cell is to be operated. How does this happen? Well, by the 1940s, it had been well established that proteins are the chemical workhorses of life. Let me review what we talked about back in lecture 46 about proteins. Remember that proteins are made from amino acids, the basic building block, and you have lots of amino acids strung together. The amino acids fold up and contort, make three-dimensional structures that are very complex, and that shape, that the three-dimensional shape of the protein is critical to determine function. Remember, many proteins serve as catalysts or enzymes in biological systems. They help synthesize different molecules that are fundamental to the operation of living things. You get enzymes that digest food in the stomach, for example, enzymes that produce heat and energy in cells. They help to assemble all matter of other substances that make up our bodies. Other structural proteins like skin, muscle, ligaments, that sort of thing. These are all parts of the protein vocabulary, if you will. You have blood proteins that transfer oxygen around the body, hemoglobin. You also have the hormones, protein-related compounds that give various chemical signals to other parts of the body and tell the body when to start manufacturing key chemicals. And all these capabilities, it's the shape of the protein molecule that's paramount. You have to think about the active site and how it can attract a molecule, how it fits. So the size and the shape of that site is critical to the functioning of every protein. Now, since proteins are constructed from a very specific chain of amino acids, which folds into a compact shape, then that sequence of amino acids is critical to the correct functioning of a protein. Remember, there are 20 different amino acids you can use, and you can choose any one of the 20 in each consecutive place along that protein chain. So the selectivity is extremely important. Different sequences of amino acids fold and twist and kink differently, creating different complex three-dimensional shapes that can lock onto other molecules in these variety of ways. So a single mistake in an amino acid sequence, that's called a mutation, may cause the protein shape to be altered, and it may fundamentally change the way that protein can follow its function. A good example is sickle cell disease, in which a single amino acid mistake, a valine, substitutes mistakenly for a glutamic acid in that sequence. And it means the protein folds up wrong, and the hemoglobin doesn't operate correctly in people with sickle cell disease. Those red blood cells are distorted and deformed as a result. In a few lectures, we'll look much more closely at these genetic diseases and how they occur, and also how they may be able to be cured. Studies of the genetic behavior of cells in which certain enzymes were mutated on purpose revealed that one gene, one set of coded information, corresponds to one specific enzyme or protein. Now, that's a fundamental idea, that a single piece of genetic information corresponds to one of these molecules that serves as an enzyme or has a structural role, or it works as a hormone. So we're getting closer to the molecular basis of genetics by recognizing that what the information that's passed from one cell to another does is make proteins. And then proteins make all the other chemicals you need in your body. It was the behavior of chromosomes and understanding by watching chromosomes that illustrated how Mendel's ideas of atoms of inheritance were actually not an abstraction, but a real physical presence. The chromosomes and segments on the chromosomes represented these genes, these atoms of inheritance. Each chromosome, it turns out, is a long structure. It's a long molecule, a long collection of atoms. And on that long chain of molecule, there are a series of genes. There are a series of separate coded instructions, each of which describes how you build a specific protein. Now, this is the reason why Mendel's fourth law was somewhat in error. Strictly speaking, it's the chromosomes that act independently of each other, not the genes. If you have two genes that lie on the same chromosome, they're not going to be completely independent of each other because the chromosomes themselves get passed off, not the individual genes. So Mendel was quite fortunate in his selection of pea plants and in the selection of the traits that he actually used in those experiments. Now to the second part of this lecture. Of what chemical substance are chromosomes made? Studies of the chromosomes under the microscope revealed all sorts of characteristics that led to an understanding of Mendel's laws in terms of chromosome behavior. For example, chromosomes are now seen to carry the atoms of inheritance that are passed from parents to offspring. Half of the chromosomes come from each parent. That's part of Mendel's laws too. Yet, these studies at the cellular level could only go so far in elucidating the mechanics and the mechanism of genetics. They couldn't show, for example, how a gene could make a plant tall, or how could it make a plant short. Ultimately, then, scientists had to isolate the molecule that carried genetic information and understand the detailed structure of that molecule. The first step was to purify and to identify the chemical substance that makes up chromosomes. And this is just a simple process of chemistry. What you have to do is pulverize cells and then extract the chromosomes in a process of centrifuging, first staining those chromosomes so you could see the color, and then centrifuging lots and lots of cells, which separated out the different parts, the different chemical components of a cell by their density. And you get a band that was concentrated in chromosomes, and you do this over and over and over again until you had enough chromosome material to give you that chemical. And then you'd analyze those chemicals using standard techniques of organic chemistry. And what they found was that chromosomes were shown to be made of two different kinds of molecules, partly proteins and partly a molecule called deoxyribonucleic acid, or DNA. Now, proteins were well known as essential building blocks of all life, but the role of DNA was totally unknown. It was uncertain what DNA would possibly do, and nobody knew what the structure of DNA was at that time either. So most scientists at the time suspected that it was the proteins that carried genetic information. After all, you have a chain of amino acids. That's like a chain molecule with 20 different letters in it. And each of the amino acids of those 20 essential amino acids could be considered one letter of an alphabet. And so you could actually carry information on a long protein molecule. That made a lot of sense. New insights were provided in the early 1940s when Canadian biologist Oswald T. Avery, who lived from 1877 to 1955, managed to isolate and purify genetically active material. And what Avery found was that the genetically active material was pure DNA. That discovery at first was controversial, and it caused much more intensive study of this DNA, this deoxyribonucleic acid, which nobody was really sure what it was or how its structure worked. What they found was that DNA contains 3 molecular building blocks. It contains a 5-carbon sugar called deoxyribose. It also contains phosphate molecules. That's a phosphorus surrounded by 4 oxygens. And it contains one of 4 different bases called adenine, cytosine, guanine, and thymine. Those are abbreviated with 4 letters, A, C, G, and T. Now, the biochemist Erwin Schargreff found that DNA displays characteristic ratios of these molecules as well, these building blocks. The deoxyribose, the phosphate, and the bases always occur in a 1:1:1 ratio. Furthermore, the bases show an additional interesting trend. The amount of A always equals the amount of T. The amount of C always equals the amount of G. So we have these curious equivalencies. And so in figuring out the structure, you have to bear that in mind. But the meaning of these ratios were going to remain unclear until the actual molecular structure was determined. This was done by purifying DNA and then growing crystals, an incredibly difficult process, making crystals of DNA. Then the British crystallographer and chemist Rosalind Franklin, who was born in 1920 and died in 1958, only at the age of 38, obtained the first X-ray photographs of DNA. This is in the early 1950s, and this was a breakthrough set of experiments to actually get X-ray diffraction measurements off a crystal of DNA, which was the key to understanding its structure. What she found, in fact, that there were 2 different forms of DNA, one that formed at higher humidities, probably one that had higher hydrogen bonding, and so the fact that there were 2 different crystalline forms gave additional insight as to how this structure might occur. She suggested that the phosphate molecules of DNA must somehow occur on the outside of the molecule and form part of a building block, a kind of framework for the whole molecule. She also suggested that the structure had some kind of helical aspect, like a spiral staircase, but she wasn't sure exactly the details of that. In 1952, building on the Franklin studies, James Watson and Francis Crick solved the astonishingly beautiful DNA structure. Watson, James Watson, a brash young American, he was in a postdoctoral fellowship at the Cavendish Laboratory at Cambridge University. Crick, a theoretical crystallographer, he was at the laboratory and much more reserved, much more urbane in the British sort of way. Their story is told in a vivid, a compelling, and idiosyncratic detail by James Watson in his best-selling account, The Double Helix, and I strongly recommend that book if you'd like to get more of an insight of what science was like in Cambridge in those early 50s. Watson and Crick's brilliant deduction, which was arrived at as much by inspired guesswork as any sort of systematic method. They tinkered with models, they sort of built structures in their spare time and sort of saw how they fit together. But they eventually came up with an astonishing structure, something like a twisted ladder. What they found was that you could think of this structure very much as you would just an everyday wooden ladder. It had 2 side supports. And those side supports were made of alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, going up and up and up on both sides of this ladder. Then you had the rungs of the ladder, and these rungs were made of base pairs, and this was the key discovery. It turns out that you have pairs of bases that fit exactly together. A fits with T, C fits with G, but A never fits with C or G, T never fits with C or G, and so forth. You have only 2 possible pairs, AT or CG. And those form the rungs of the ladder. That's why you always have a 1:1 ratio of A and T and a 1:1 ratio of C and G. Then you have this ladder-like structure, but the whole structure is twisted into a helical shape, the double helix, because it's a 2-sided polymer. Indeed, it's called sometimes a double polymer. This DNA strand can be virtually any length. It can be up to billions of nucleotides long. Just a long continuous ladder, a double polymer structure twisted on itself into a helix. Watson and Crick immediately recognized the power and the beauty of this elegant DNA structure. It lies in its ability to store and to copy vast amounts of information. And that's the key in genetics, isn't it? It's passing information from one generation to another. The 4 bases, A, C, G, and T, are a 4-letter alphabet, the genetic alphabet. They're capable of conveying any amount of information. depending only on the length of the molecule that you put together. Lest you think that 4 letters is too few to convey information, remember that our modern computer age, the digital age, is based on just 2 letters, if you will, 0 and 1. So 4 letters is actually kind of a luxury in terms of conveying information. This double helix configuration with complementary bases opposite each other also gives you a way of duplicating information, because each side of the structure contains all of the molecule's genetic information. Look at more detail how this works. First, how does DNA carry a message? The role of DNA, which is to carry biological information, is that it has the 4-letter alphabet. Now here's an activity you can actually do with children if you ever want to. Imagine getting some beads, 4 different colored beads, for example, or 4 different shapes. You can create your own alphabet, your own equivalents to the 26 letters of the alphabet. You're going to need 3 beads to make each of those letters. And then you invent a code, for example, that only you and your children know. You can then create various bracelets or necklaces of the beads in different orders, and you can send secret messages back and forth because only you know the code of those 4 letters. It's a lot of fun, and it's a great way to indicate to children how the genetic code really works. In exactly the same way, DNA's 4 molecular letters can convey any conceivable message. Now also think about how DNA copies itself. To do this, the cell needs several things. First, you need to have an enzyme, that is a protein, that unzips the DNA double helix. This is a specially designed protein that has to be in every single cell, because every cell has to be able to duplicate itself. So just before the cell divides, the DNA has to duplicate it. The DNA is essentially unzipped, just like a zipper, by this protein, and you expose the 2 halves of the double helix. You have 2 single strands. The cell also has to have vast numbers of unattached nucleotides, that is, the sugar-phosphate-base combinations, to fit in. If there's an exposed A, a T comes in to fill the spot. If there's an exposed C, a G comes in and fills in. And so where before you had a single double helix, now you have 2 double helices. You've made a copy of the original just by unzipping it and exposing the 2 sides of the original double helix. You end up with 2 identical chromosomes where before you had only 1. In their landmark paper, it was a 1-page article in Nature in 1953, Crick wrote in the final sentence, and this is a quote, "It has not escaped our notice that the pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." A wonderful understatement, if there ever was one. And for their discovery, Watson and Crick won the Nobel Prize in 1962. Harvard biologist E.O. Wilson has commented on the significance of this discovery. He said, quote, "It is hard to imagine the impact the discovery of the structure of DNA had on our perception of how the world works. Reaching beyond the transformation of genetics, it injected into all of biology a new faith in reductionism. The most complex processes, the discovery implied, might be simpler than we had thought." In summary, the nature of the genetic process was elucidated by studies of chromosomes, particularly by their behavior during the process of mitosis, that is simple cell division, and also in meiosis, that is sexual reproduction. In these events, chromosomes duplicate, they divide, and they recombine in ways that are exactly analogous to Gregor Mendel's atoms of inheritance. Early in the 20th century, the chemical substance of chromosomes, that is DNA, was extracted and identified. And then in the early 1950s, Watson and Crick, with the help of Rosalind Franklin, determined the atomic structure of DNA. The power of the double helix lay in its simplicity. It's a ladder-like structure with variable rungs that could at once store and duplicate vast quantities of information. Scientists had found the book of life, but a tremendous challenge lay ahead. They had yet to learn to read that genetic language, and that's the story of the next lecture.